Hi everyone. So next up we have chapter 5, which is cell respiration and metabolism. And you've probably talked about this a bit in one of your other classes, right? So some of this should be review, but this is obviously a really important topic because we have to think about our bodies using and metabolizing nutrients, okay? And we know, we talked about last week, that there's 3 major nutrients used by the body. We have carbohydrates, lipids, and proteins. And so how our body uses these has different mechanisms in which to do that, and then certainly different jobs for these kinds of fuels too. Now, when we say metabolism in sort of a colloquial context, people think of metabolism as like digestion, maybe, or how fast you burn energy. And that's sort of true. When we talk about metabolism physiologically, it's divided into 2 parts, okay? We have catabolism, which is breakdown of nutrients for energy, ATP. So taking nutrients and turning them into energy, turning them into ATP, and building blocks. So breaking stuff down and turning them into other stuff. Anabolism then is using energy and building blocks to build new products. So catabolic is breaking things down. Anabolic is building things up. You can think of something like anabolic steroids, meaning to build, okay? And so we think about breaking down carbohydrates, carbohydrate catabolism. Glucose is our major energy source of the body. It's our primary blood sugar, and it's what we use most often to get energy. Okay. So we're going to talk mostly about this, mostly about the use of glucose for energy, which is aerobic respiration, right? So aerobic respiration is this process that cells use, also called cell respiration, that we're going to take primarily glucose and turn that into ATP, energy. Now, we'll talk later about how glucose is not the only thing you can put into aerobic respiration, but we're going to start here, okay? So we have 3 processes we're going to talk about today in aerobic respiration. There's glycolysis, the Krebs cycle, or sometimes called the citric acid cycle, same thing, and the electron transport chain, which is abbreviated as ETC. So we'll talk about these 3 processes today that get us from glucose to ATP. And here is our major formula that I want you to know. And I do want you to memorize this because this is the basis of all this stuff. It's pretty important. So here's glucose, C6H12O6. When you take 1 molecule of glucose, you add 6 molecules of oxygen, and that reaction of cell respiration gives us water, carbon dioxide, 36 ATP, so 36 ATP molecules per 1 molecule of glucose, and heat as a byproduct. Okay, so in biological systems, heat is a common byproduct, and that heat can't be captured and used again for any kind of energy, but it is used in the sense that it can affect our body temperature. We're thinking about things like body temperature, homeostasis, that comes into play, okay? So body heat is produced here. And so these 3 processes, glycolysis, Krebs cycle, and the ETC, get us from glucose and oxygen to water, carbon dioxide, ATP, and heat. Okay. So here's looking at this glucose in the plasma. I said that glucose is our primary blood sugar, and glucose is coming in from the digestive tract. So when you eat a meal, and your body digests, you're digesting things in your stomach and small intestines, right? So when you digest carbohydrates, that glucose then is transported from the digestive tract into blood plasma. Okay, so it's floating around in the blood plasma. You can also get glucose from the liver because we mentioned in lab that we stored glucose in the liver as glycogen. So glycogen is a big glucose compound that can then be broken down to put more glucose into the blood. Okay, and then glucose circulating in our blood. And glucose is going to be at a homeostatic level in our blood. It does go up and down, right, just like any other homeostatic system, but the set point in our blood makes us sure that we have enough to fuel our bodies. Okay? And so then what happens is your cells, this can be any cell that has a mitochondria in it, cells take that blood glucose and they transport it into cells and then put it into these 3 processes. So here's glycolysis, here's the citric acid cycle, and then here's electron transport. And those three processes together is aerobic respiration. We'll talk a little bit about anaerobic metabolism too. But the difference you can look at now is that anaerobic metabolism is done without oxygen. Okay. And it's way less efficient than aerobic respiration. Aerobic respiration takes us from glucose all the way to, we said, 36 ATP per 1 molecule of glucose. Okay, so what's happening? Let's break this down. Glycolysis, that process happening in the cytosol of cells. You take a glucose molecule that has 6 carbons, and you break it into 2 pyruvic acid, so 2 3-carbon molecules. So this is glucose catabolism because you're breaking down glucose into 2 molecules. Additionally, in this process of glycolysis, we have 2 NAD+ molecules that are turned into 2 NADH molecules. The pyruvic acid coming out of this process moves on to that Krebs cycle, while the NADH go to the electron transport chain. So these NADHs are electron carriers. They're going to take electrons and bring them to the ETC. And that'll become important later. In the process of glycolysis, there are 4 ATP made, but you need 2 ATP to invest in glycolysis in the first place to get it to run. So you actually only get 2 ATP out of glycolysis. Okay, so it gives you a little bit of ATP. So our final net product then for this is 2 pyruvic acid, 2 NADH, and 2 ATP net. Okay. There is no oxygen required for glycolysis, so this process is anaerobic. So we haven't invested any oxygen yet. Okay, so this is glycolysis in the chemical form. You do not need to memorize this at the intermediates. I want you to focus on the carbons where you have glucose here, right? 6-carbon molecule. And here is just showing where the 2 ATP are invested, right? We have a bunch of intermediates that you can ignore. And then through a bunch of steps, we break that glucose into 2 3-carbon molecules of pyruvic acid. And along the way, we get out 2 NADHs. And so those are going to go to the ETC later on. And you see we get out 1, 2, 3, 4 ATPs, but we invested 2 here. So we get net 2 ATP. So focus on the carbons, movement of and production of NADH and ATP. But you don't have to memorize these intermediates. Okay. So that process is glycolysis, taking glucose, splitting it into 2 pyruvic acids. If there's no oxygen available, okay, then the pyruvic acid is made into lactic acid, ethanol, or other product, and that process is fermentation. So in our muscles, this can happen. So in muscles, when we don't have enough oxygen available, pyruvic acid can be turned into lactic acid. And you might have heard some mostly myths about lactic acid and things like soreness. And while lactic acid does build up in muscles, it doesn't seem to be related to how sore you get after a workout. Okay, so that's one of those kind of myths that because they correlate happen to come together, but that's not really true. And what happens here is not that you get any more ATP here. Remember, we got 2 ATP turning that glucose into pyruvic acid. But the reason why this happens when there's no oxygen available is that there's no electron transport chain for those NADHs to go to. And you need NAD+ to run glycolysis. So what happens then is as pyruvic acid is turned into lactic acid in our muscles, the NADH is turned back into NAD+, and that can then run glycolysis again. So it's a way to recycle the NAD pluses to be able to send them back into glycolysis. So it's a way to keep glycolysis going, which gives off 2 ATP, right? So it's not super efficient, but it works. And so bacteria do this too, right? Where they're going to be relying almost entirely on anaerobic respiration in some cases. Okay. So we also use this process in bacteria and yeast to make a whole bunch of different products, right? So our muscles make lactic acid, but some of these bacteria and organisms make other stuff that we like, right? So cheese is made as a fermented product. Lactic acid can be used to make cheddar cheese and yogurt and soy sauce. Wine and beer are products of when ethanol is produced in the fermentation process. Products like acetone, isopropanol, so things that we use for nail polish removal and rubbing alcohol are there too. And then acetic acid and vinegar is also a product here. So these are all different examples of ways in which these microorganisms have done the anaerobic respiration. So they've done glycolysis, right? Here, glycolysis. And then they turn that pyruvic acid into other products that we then use and like. Okay. If you want to make a lot of ATP, you can't rely on that only. So, yes, our muscles do a little bit of fermentation. Mostly, though, once pyruvic acid is produced from glycolysis, that pyruvic acid is put into the Krebs cycle. Okay? And this is happening in the mitochondria. Okay? So the pyruvic acid is turned into something called acetyl-CoA, and then the acetyl-CoA is put into the Krebs cycle. So there are 8 steps in this. You don't need to memorize the steps. I want you to focus on what's happening here, okay? So you're putting pyruvic acid, turning that into acetyl-CoA, putting acetyl-CoA into this cycle. And this is going to happen twice per glucose molecule, because remember, you went back there and you made 2 pyruvic acids out of glucose, right? So we're making 2 3-carbon molecules here that we're putting into this cycle. This process is actually also anaerobic. We don't need oxygen quite yet. We will in just a minute. And what's produced are 4 carbon dioxide, 6 NADH, and 2 FADH2. So those are more electron carriers that are going to go to electron transport chain, which is our next step, and 2 ATP. So this process gives you a little bit of ATP. These are the products you get from those 2 spins. So putting those 2 pyruvic acids via acetyl-CoA into the Krebs cycle. So 1 glucose molecule gives you 2 spins of the cycle. And here's what a mitochondria looks like. You've got an outer membrane, and then there's a second folded membrane on the inside. Those folds are called cristae. The matrix is the internal space, and then here is the intermembrane space. So you see that this cycle is taking place in the very interior of the mitochondria. Okay, so here's the whole thing. You don't need to memorize this. What you need to look at is, here's acetyl-CoA coming into the Krebs cycle, okay? And carbons are moved around and changed. But what's important, I want you to see that it's a cycle, because what's happening is the acetyl-CoA is combining with a product of the previous turn of the cycle to make the first step in this cycle. Okay? So think about that. Acetyl-CoA coming in, combining with this oxoacetic acid to make citric acid. That's the cycle. And look what's coming out as these reactions take place. Okay? You got an NADH here, NADH, FADH2, there's an ATP, right? So this is one turn of the cycle. You put a second acetyl-CoA in that you also got from turning pyruvic acid into acetyl-CoA. And now you get that happening again. So if one turn gives you 3 NADHs, 1 FADH2, and 1 ATP, 2 turns that you would get from 1 glucose molecule gives you 6 NADH, 2 FADH2, and 2 ATP. Okay. So now we can move on to electron transport chain. And so this is after glycolysis, after the Krebs cycle. Now we have the ETC, and here's where you get most of your ATP then from glucose. Okay, so there's these pumps. Here's one here, one here, one here. These are electron pumps. Okay, so they're pumping. And we have this really important enzyme here called ATP synthase. So what's important is that you note that these pumps are pumping into the intermembrane space. So see all these H pluses here. And what's happening is that they are creating a high concentration of hydrogen ion out here. Okay, what happens, think back to basic biology, what happens when you have things at an area of high concentration and things at an area of low concentration. Stuff diffuses. Okay, so we have these pumps powered by electrons creating a high concentration out here. And then the only way these things can diffuse back into the matrix is through the ATP synthase. So the pumps create the concentration gradient, and then as the hydrogen ions travel back into the matrix, they spin the ATP synthase. It's a literal spinning of this enzyme that creates ATP. Okay, so I want to remind you that these electrons came from those earlier steps, right? They were transported here by NADH and FADH2 that we got from glycolysis and the Krebs cycle. They came here. Now they're doing this job of powering the pumps. And then once the concentration gradient is set up, now the hydrogen ions come back in through the ATP synthase. They're diffusing down through this channel, through this enzyme, and it spins and creates ATP. Okay, and I told you in the beginning that 36 ATP come from one glucose molecule. 2 of those come from glycolysis. 2 come from Krebs cycle. So it's 4. Now we have 32-ish, 32 ATP coming from one glucose molecule just from the ETC. So this is the most bang for your buck, where you get most of the ATP from that single glucose molecule. Okay, so in the inner mitochondrial membrane, now we're requiring oxygen. Oxygen is required because it's a final electron acceptor. It's accepting all those electrons that were transported there earlier in the process. Okay, so without oxygen, this can't happen. Without oxygen, you can't have the most efficient production of ATP. Okay, so I want you to focus on following the ions. Okay, the transfer to the membrane space we talked about, and the passive diffusion through ATP synthase. Okay, and we said one glucose molecule gives us about 26 to 28, up to 32 ATP per glucose molecule. There's another video link on here too. That's really good to watch. There's lots of cartoons and videos of these things on YouTube, but it's a particularly good one that you can check out too to learn more. Okay, so the ATP yield kind of differs depending on how you measure it, but you're getting somewhere between 30 and 38 ATP per glucose molecule. Okay, so at the end here, they're getting, we're making also water from this. So water is a byproduct of the cell respiration. Okay, so recap. Again, here we've got glucose coming in glycolysis. It's getting turned into 2 molecules of pyruvic acid, also called pyruvate. That gives you a little bit of ATP. Taking that pyruvic acid and putting it into the Krebs cycle in the mitochondria gives you a little bit of ATP. The electron carriers now, so the NADH from glycolysis and the NADH and FADH2 from this Krebs cycle, power this electron transport chain. Sometimes it's called oxidative phosphorylation in the text as well. Same process. It powers that through the ATP synthase, right, to make a lot of ATP. And that whole process is aerobic respiration. It's aerobic because oxygen is required for this last step where you get the majority of the ATP. Okay. So what has been interesting is that there's some drugs that inhibit the process of glucose metabolism and result in a decreased production of ATP. So I want you to think about this now and also think about it as an option for your discussion, is to think about the following poisons and how they inhibit ATP formation in the mitochondria. This is an at-home thought question. There's cyanide. Cyanide inhibits the 3rd pump in the ETC. Okay, so I want you to think about how that's going to affect this whole process. There is rotenone. which blocks NADH dehydrogenase. This is actually an insecticide used on plants. Okay. And there's FCCP, which makes pores in the mitochondrial membrane. So think about how that might affect these metabolic processes. Okay. And we just talked about glucose, but I want to point out that you can have multiple things going into glycolysis and going into this aerobic respiration cycle besides just glucose. So of course, the polysaccharides in our food that we eat, we break down into glucose, and we can put the glucose directly into glycolysis. Fats, remember we have fatty acid tails in our fats. You can get conversion of the glycerol into something called PGAL you can put into glycolysis. Fatty acids can be turned into acetyl-CoA, and that can enter the Krebs cycle directly. Proteins can be broken down into amino acids, and you can put those in as pyruvate, acetyl-CoA, or into the Krebs cycle directly. And so you don't need to know the names of these things, but just realize that it's not all about glucose, right? Anything you eat, you can break down or convert and put somewhere into the respiration. And not everything you eat. There's some things like insoluble fiber. Insoluble fiber doesn't get broken down, comes through us. It's good for gut health because it helps us poop, basically. But anything you can get energy from, you're getting energy from by putting it into one of these steps of this aerobic respiration. Okay? So that's breaking stuff down. Let's talk a little bit about building stuff up. So for carbohydrates, building carbohydrates, we said that one important thing to build is going to be glycogen, right? So we have our primary blood sugar, glucose, floating on in our blood. Sometimes, a lot of times, we have extra. Okay, well, what do we do with that extra? Well, we could just, theoretically, you could pee it out or something. But why? Hang on to that energy, use it later. So this is a major important component of us being mammals and animals in general, is that we can store food, we can store energy, or we can store an energy source, right? So what happens then when you have extra blood glucose is glycogenesis. So genesis meaning birth or building, right? And glyco is glycogen. So you're building glycogen, making glycogen from excess glucose in your blood. This is controlled by insulin. And glycogen is stored in liver and skeletal muscle. So this is great because then later on when you need that glucose source and you can't eat right then, now you have something you can break down and use for energy. Glycogenolysis is the opposite. That's the breaking it down. So then when you want to break down glycogen to use it as blood glucose, that's called glycogenolysis. And lysis is a word that means break. We're going to hear that a lot in cells and in this class. Okay, you have lysis of blood cells, but essentially means breaking. So glycogenolysis, glycogen breakdown. Okay. And that's controlled by glucagon. Okay, so if you need more blood glucose and you need to pull it out of storage, you break down glycogen with the help of the hormone glucagon. Okay. So here's this process. Here's glucose. Okay. Glucose coming in from your blood. Just showing you this pathway you can put it into glycogen or go to glycolysis too, right? So start that cell respiration cycle. Okay. For lipids, we can actually build fats from sugars. And remember acetyl-CoA, which is that product of essentially glycolysis and then turning that pyruvic acid into acetyl-CoA, can be transformed into lipids. Okay. So we can make cholesterol, triglycerides, and fatty acids in our bodies. This is great. These are important components of cell membranes, energy storage, all kinds of important components, right? And this is termed lipogenesis. So lipo, fat, creation, making fat. We mostly store triglycerides as white adipose tissue, and those are going to be long-term energy storage deposits. Whereas we have a special kind of tissue called brown adipose tissue that's found mostly in newborn humans and is used for making heat. For some other animals, they use brown adipose tissue a lot, like bears and bats, particularly like hibernating kinds of animals will use it because it is a really good heat producer. And so if you're doing something like you don't have a lot of availability to eat at the time, newborn humans, they need to be trying to keep themselves warm when they are really, really small. And so this brown adipose tissue, sometimes called BAT, is really important. Okay. Breaking down fats for energy is lipolysis. Okay. Breaking down lipids into glycerol and free fatty acids. Remember, we can put some of these components into that cell respiration pathway at different points, right? We can take glycerol and turn it into pyruvic acid and turn that into glucose, okay? We can take fatty acids and convert them into acetyl-CoA to put them directly into the Krebs cycle. And excess lipolysis, so lots of fat breakdown, results in ketone bodies. Okay. You might have heard, we talked about, I think before too, this ketogenic diet where you're trying to make your body make ketone bodies essentially because the idea is to promote fat breakdown, right? But the problem is that ketone bodies can cause blood acidosis. So it can cause your blood to become too acidic, to lower in pH too much, and that can be very dangerous. So we'll talk about that a lot more later in this class when we talk about acid-base balance. But extremely fast weight loss and excess lipolysis can definitely be dangerous because essentially it throws off your blood's pH. Okay. So talking about proteins, proteins are usually not used for energy. They can be, but they're usually not. Instead, we use sugars first. So your body has its kind of mechanisms to do that first, right? Most readily available is going to be that glucose. So your body uses sugars first, then fats, and then proteins if it has to for energy. Primarily because proteins are going to be doing other stuff, right? Proteins are going to be in muscles, proteins are going to be for other structural components, things like that. The proteins you eat are broken down to amino acids, and you use those amino acids then to make new proteins, right? To replace older used proteins. So you're thinking about making new skin cells, making new blood cells that have proteins. You need to make new proteins a lot. And so you need amino acids in your diet in order to create more proteins. Excess protein is not stored. So any of these kinds of really heavy protein diets and building up protein shakes, things like that, they're really not that helpful because once you can't store it, right? You have to keep consuming protein in order to have enough amino acids. And so they get released in urine as urea. And so what happens is people on very high protein diets tend to have sometimes have kidney problems because they're producing so much urea from the excess protein and your bodies can't use all of it. So if needed, if you're kind of at this last ditch result here, amino acids can go into pathways to make energy. Okay, so you do have those three options then for energy. Okay, so just another visual of this process, right, where you can have multiple different conversion processes that turn these things into things for the Krebs cycle. Okay. And so just briefly, I want to talk about this relative importance of different molecules in the blood with respect to the energy requirements of organs. Our brains are the glucose hogs. So brains need lots and lots and lots of glucose, and that's where the majority of our glucose goes. But we do need glucose to fuel other things too, like skeletal muscles, liver and heart tissue, right? Fatty acids are not really that important to the brain as far as we know, but can be more important for muscle, liver, and heart tissue. And you can see lactic acid having a slight effect in requirements in liver and heart tissue, but not in muscle, and then ketone bodies as well. But the one I want to mostly point out is this glucose because your brain is going to be using the majority of your glucose. Okay, and that's it for today. That's our brief foray into cell respiration. So please review this stuff, focus on what I told you to focus on in this video. and definitely study these different mechanisms to think about how we're using food for energy. See you next time.